I'm Sarah Fitzsimmons. I work for the American Chestnut Foundation out of our Penn State Partnership Office. I'm the Director of Restoration and your humble host of Chestnut Chat, which we hold now every third Friday of the month through 2021. Thank you for attending our first one. Robert said he'll still wear pants. That sounds good. I'm glad glad some people are. I think that's all the housekeeping stuff. Today we have Amy Metheny, who's joining us from WVU. She's been working with Chestnut Blight and Hypovirulence there as a research associate for 2 years, 3 years?
Yes, officially, but I've been in the lab for 8.
That's right. I know I mentioned this earlier, but just to repeat, we had excellent sessions of Chestnut Chat. I really enjoyed doing that through 2020. And the number one topic that I had people ask about was doing something on biocontrol. And so I'm thrilled to have Amy here to talk about biocontrol. Before we turn that over, I'm going to have Lisa, who is our fearless leader, talk a little bit about opening up Chestnut Chat, and then she can pass it on to Amy when she's ready.
Okay, thank you, Sarah. Welcome everybody. Happy 2021. I know we were all anxious to get out of 2020, even though 2021's off to a bit of a rocky start, but we won't go there. The Chestnut mission is solid and going forward. I just want to thank all of you for joining us today. The engagement of Chestnut Chat has really blown us away. I think Sarah did some analytics on it. Maybe she can have those handy at the end of the chat today when Amy's done. But it was really a wonderful way to keep our community engaged, because we love to share ideas, stories, discoveries, and that's what's happening today with our first Chestnut Chat with Amy. She's a really inspiring young scientist, and I'm so glad to have her part of our Chestnut community. And to all of those, our work wouldn't be possible without the generosity of all of our donors and members, and our end of year campaign was really, really strong. So thank you so much to all of you that either donated or told your friends about the Chestnut Foundation. We can go forward without constricting any work that we're doing, which is really important, all while maintaining safety protocols, especially in the field. We haven't reopened the Asheville office where I'm based yet. We all go in, we take turns. My day is Wednesday. So we don't, endanger each other unknowingly. So we're still being pretty strict about that. But we really hope that, by the summer, maybe we can all gather, and we sure hope we can gather October 30th in Asheville for our fall meeting. So let's just keep the fingers crossed out there. And with no further ado, thank you so much for joining us, Amy. Take it away.
All right. So hi, everyone. My name's Amy Metheny, and I am a research assistant at West Virginia University studying chestnut blight. I've been here for about 8 years, but in my current position, I've been here 2 years. And I'm here to talk to you today about hypovirulence and chestnut blight. So I'm going to try and give a very kind of basic, and please let me know if you can't hear me. Sometimes my laptop audio gets a little funky, but I'm going to try and give a very basic presentation to kind of give you an idea of how chestnut blight works, how hypovirulence works. And this is something that can be a little off-putting at first, because there's a lot of moving parts of things that maybe we're not used to dealing with in everyday life. So I encourage you to write any questions you have in the chat, and we'll get to them as we can. And if there's any we can't answer, please send them to my email.
Yeah, Amy, I'm gonna just real quick, we're gonna use the Q&A module. So use the Q&A to ask questions. That way we can keep track of them. Kendra Collins, thanks for joining us. Kendra is gonna help answer, and I will too help answer some of them by typing, but we will hold them all until the end. If you just wanna tell us something, throw it in the chat, but if you have a question, use the Q&A.
Excellent.
And Amy, you sound great. I'll interrupt you if we can't hear you, but so far you're doing great.
Good, thank you. I can't tell you how many times I've got halfway through something and figured out no one can hear me. Okay, so if you're here, I'm going to assume that you have a basic idea of the American chestnut. So I'm going to spend just a little bit of time talking about the history of the American chestnut. As we know, Gifford Pinchot described the American chestnut as a very common, large, and valuable tree reaching a height of 120 feet and a diameter of 13 feet. which is absolutely insanely huge. When we think of American chestnut as a forest tree, we should think of it similar to maybe one of our oaks, because they're very closely related and they have similar growing forms. So we should think of a very tall tree with a sweeping canopy and a clear trunk. It had very beautiful, easy to work with wood, which made it a very desirable woodworking tree. So it's often dubbed the cradle to grave tree because you were likely to lay in both a cradle and a grave made out of American chestnut. It was very, very prevalent, and because of its large size and good timber form, that means that it was able to make up over 25% of hardwood timber cut in southern Appalachia. I believe that this was from a certain region in Tennessee. But people loved to use it because it produced nuts every year, as opposed to some other hard mass species like your oaks, which produce acorns every 2 to 5 years. So if we look up here in the top left corner, we can see a picture of some farmers actually sorting through these massive mounds of burrs in order to get nuts. This tree really helped people to supplement their food stores. Chestnut can be stored for a long time. You can grind it into flour, make bread out of it. You could feed your horses and cattle and pigs with the nuts. So it was something that really became a huge symbol of cultural importance, especially in Appalachia in the early 1900s and late 1800s. In the middle, this is just a depiction of a large American chestnut in Pennsylvania. A lot of people used to have gatherings where they would get together in the fall and collect chestnuts and roast them over the fire. And just, I'm trying to hit home the importance culturally to people. It wasn't just a tree that had value because you could cut it down and use it for timber. It also had a lot of importance to people's livelihoods and just cultural traditions. So the reign of the American chestnut couldn't live forever. In 1904, chestnut blight was discovered in the Bronx Zoological Park in New York City, although it was likely in the US for 10 to 20 years before its official discovery. The chestnut blight fungus is a problem, in part because it infects all castanea species, which are chestnuts, but it can also live superficially on some quercus species, which are oaks. So even eliminating chestnuts, as the early control attempts tried to do, didn't really put a dent in chestnut blight, because it can exist in our forests on other species, not exclusively chestnuts and chinkapins. When chestnut blight was first introduced, it spread at a rate of about 22 miles per year, which is very, very fast for a fungal disease. We might see something like this with insects that can move independently and fly and hitchhike on people's cars and things like that. But seeing a spread of 22 miles a year for a fungus is striking. And because of this, it destroyed millions of dollars of chestnut timber before it was really able, before we were able to get any kind of handle on it. And I just love, I just want to take a minute, because I'm a little bit of a history nerd, to talk about this official call for conference I found online. This was from 1912. And the governor of Pennsylvania was getting a lot of legislators and scientists together to discuss the danger presented by the prevalence and spread of a fungus disease of the wild chestnut tree, known as the chestnut bark disease. And in this conference, they actually initiated a cut and burn control attempt in which they foresters would go out and cut down chestnuts along the disease front and burn them in an effort to stop spread of chestnut blight, because they thought if it can't jump from continuous chestnut trees down south it won't be able to spread. But again, chestnut blight can live on Quercus species, so that ultimately was not successful. So we can see here, this little dot is New York City, where chestnut blight was first discovered. And this is a map that shows the spread every 10 years of chestnut blight. So if you look, by 1950, chestnut blight had covered the entire native range of the American chestnut. And in its wake, it left scenes like this, in which we had previously large, healthy standing trees in the forest that are just annihilated very quickly, killed within a few years, and we have a lot of dead standing timber. Chestnut today, a lot of people think that the American chestnut no longer exists. That's not true. We have many American chestnuts in our forests. They just can't get very large anymore. So as a chestnut, they do very similar things as oaks do in their growth habit, and they put a lot of energy into their root system. They put a lot of energy into having a large, healthy root system, and that's also a place they can store energy. So if an American chestnut is killed by blight, it can often use the energy that it's stored in its roots to send up shoots. And we can see this here on the left. A lot of the time when you find American chestnut in the forest today, you'll have clusters of stump sprouts that are all coming out of a place where there was once a larger American chestnut. Typically, they get to be around 20, 25 feet tall or less before they're killed by the blight ultimately. And we can see here on the right, just as an example of what we might have seen earlier on if we were to see chestnut blight sweep through when trees were larger. These are some escape trees that were planted in Wisconsin that were killed by chestnut blight a couple years ago. These are some of the biggest trees I've ever seen in the forest. So we know what chestnut blight did. We know that the American chestnut was very important and that it was taken, but how exactly does the chestnut blight fungus work? So I've broken this down into 4 really basic questions to kind of boil it down as far as I could. How does the fungus enter the tree? How is the fungus spread? What does the fungus eat? And how does the fungus kill the tree? So first, how's the fungus spread? And don't worry, I'm not going to make you memorize this crazy diagram over here on the right, but it's just a good way to kind of show what's going on in this disease cycle. So the fungus spreads itself through sexual and asexual spores, like a lot of fungi do. The asexual spores, there's a depiction here with this little curly Q. The asexual spores are produced and they just kind of ooze out of the canker. So they're very heavy, and these are typically going to be spread by water droplets. So rain hitting the spores and splashing them, or running down the stem and pulling spores with them, or by animals. So this can be insects feeding that pick up some spores on their feet or their bodies, or even things like birds. Here on the left, we see a picture of a passenger pigeon. This is a type of bird that went extinct because of overhunting by Europeans, but researchers have actually gone back and found chestnut blight spores on the feet of stuffed passenger pigeons. So, you know, birds, insects, a lot of different animals can help spread this fungus. But the number one way it's spread is through sexual spores, which the picture more to the left is. And these spores are kind of shot out of the fungus. So here we can see a canker. We see this orange tissue. This is called stroma. And we see these little things, these little lines poking out of them. And these are called parathecial necks. which isn't something you need to remember, but a parathecia is just kind of a sac that holds the sexual spores. And when they're ready, it shoots them out of these little necks that you see coming out of the bark. And those are typically going to be spread by wind. It spreads very, very well in the wind. Sandy and Agnes Sakis has some excellent papers on levels of spores in different areas in wind. And it's just the major way that they're able to get around. So number 2, how does the fungus actually enter the tree? Well, the chestnut blight fungus doesn't have a way to penetrate the bark. It is not, unlike a lot of other plant pathogens, it doesn't have a special structure or special compounds or anything that penetrate the bark. The fungus relies on finding a wound on the tree. This can be due to just things that happen. Here on the left, we see a lot of cankers arise from where there are branches. So here we see 3 cankers all coming out of areas where there's a branch or a branch stub. And that's because when the branch is attached to the trunk and it flexes with the wind or with a deer walking by, or it's rubbing against another branch or something like that, this can create tiny little wounds in the bark. And if a chestnut blight fungus spore lands on that wound, it can infect the tree. Animals are a big factor. Here in the middle, this is actually one of my trees in Savage River State Forest in Maryland. I had a bear rip all of the, or many of the branches off of my tree to get to the chestnuts. And this was eventually just covered in one massive canker. You can also have animal damage by deers rubbing the tree or insect feeding damage. Even like chipmunks and rabbits will chew on bark sometimes. And that can be a mode of entry. And also there are lots of man-made mode of entries. And this is a photo from Linda McGugan. And here we see just a piece of flagging that was left on the tree too long and cut into it. And that can provide a good place for a canker. So we need to make sure that we pay attention to our trees, to flagging and landscape fabric, tree tubes, things like that that could create little wounds. Now, number 3, what does the fungus eat? Because this is the whole reason the fungus wants to attack American chestnut, right? Is because it's hungry, it needs a food source. So I'm a tree nerd. I did my undergrad in forestry, so I could really talk about trees all day. But I'm gonna try and keep this as accessible and not boring to you as possible. So what the fungus does, once it enters the tree, it wants to eat something very easy to break down and very nutritious. So when we look at a tree, at a column of a tree, only the very outside little bit is actually physically living. And that's this area called the vascular cambium. This is where all growth is most growth is taking place. It's making xylem and phloem, which are very important to the tree. And all this middle heartwood is just kind of hanging out in a state of neutrality, I guess. It's just not really doing anything. It's hanging out there. It's not technically dead, but it's not really alive. So when the fungus enters the tree, it wants to eat this yummy, delicious vascular cambium, which is easy to break down. And since that's where all the growth is happening, that's the most nutritious portion. So if we look here on the right, we can see a canker, and we can see that this brown area, this is the canker, and that's the area that the fungus is actually using for food. Now, the fungus can live on dead tissue, and it can eat dead tissue, but it does really like this fresh, nutritious, yummy stuff. So how does the fungus actually kill the tree? Well, because the fungus is eating the yummy, nutritious, living portion of the tree, it's stopping the fungus's ability or the tree's ability to create new tissue. And it's also starting to break down some of the very important transportation structures in the tree. So the tree is built up of a lot of basically straws that send water and nutrients throughout the tree to keep it alive. And once the fungus breaks those down, the tree can no longer transport water and nutrients, and so the portion above the canker dies. This is another photo sent to me by Linda, and you can see that above this canker, this is dead, and this canker has ultimately gone around the entire outside portion of the tree and broken down all of that tissue. So that means that nothing above that point is surviving. However, like we talked, the roots are still healthy. The chestnut blight fungus does not survive very well in the soil because it's a weak competitor and it's outcompeted in the soil. So we can see here, this tree, although the main stem is dead, the roots are still living and they're sending up these nice, healthy, happy shoots, which will probably grow for a good long while and then ultimately succumb to chestnut blight. But there is hope. I know that it sounds really scary and horrible and devastating, and it is, but there's hope that we can beat the chestnut blight fungus. So this phenomenon called hypovirulence was first discovered in Italy on European chestnut in the early 1950s. So these mycologists, who are people who study fungi, were walking around in the woods and they saw that some of these European chestnuts, which are also susceptible to chestnut blight, had cankers that didn't look sunken and orange and angry like a lot of the cankers would be looking in the forest. They saw cankers that were starting to swell. The tree was having some sort of immune response. There we go. It's they were having some sort of immune response. So they're swelling and they're starting to kind of grow over these cankers and heal over that area. And obviously this is something that they thought needed to be investigated because they just looked so vastly different. So when they sampled these cankers, they saw that it was Cryphonectria parasitica, which is the fungus that causes chestnut blight, but it looked really strange. It was white. So they kept doing research, they kept doing experiments, and eventually found out that this white version of the chestnut blight fungus had a virus. So it was sick. And we call these hypoviruses. So up at the top is a picture of a sample from a canker that was killing a tree. It was growing quickly, producing a lot of spores, and it's orange. Chestnut blight is very, very characteristically orange. But on the bottom, we see a sick individual from a canker that was healing over. It's white, it doesn't grow very quickly, and it doesn't produce very many spores. So I'm going to take just like one brief minute to hammer home the terminology because it can get really confusing. So we're talking about a virus and a fungus and a tree. So when we talk about hypovirulent isolates, we are talking about the fungus, okay? Because when the fungus becomes sick, it has reduced virulence. So it is less able to kill a chestnut tree. So to make it easier, kind of think a hypodermic needle and a virus. Okay, so hypodermic needles go under your dermis. Hypo means under, dermis is your skin. Virulence. So hypovirulence is the reduced virulence of the fungus. And a virulent isolate of the fungus is virulent. It can kill the tree quickly. It produces a lot of spores and it's very dangerous. Okay, so hypovirulent isolates of Cryphonectria parasitica produce slower growing cankers with reduced asexual spore production. So here on the left, we can see this is a canker that was started by a hypovirulent isolate, a white sick one, and we see how tiny this is. And on the right, we can see a very large canker initiated by a virulent version of the chestnut blight fungus, which is doing its absolute best to kill the tree. And this gets into an issue that I'm not going to spend a lot of time on because we could do a whole nother seminar on it. But there are some barriers to spreading this hypovirus. We can't just go out with a backpack sprayer and spray it around in the forest, or, you know, drop it out of an airplane or something like that. This virus can only live inside of the chestnut blight fungus. So it's not something like the flu or COVID where you can sneeze and it lands on a surface and the virus is still living. This hypovirus would die if it was extracted from the fungus. So we have to give the virus, we have to spread the virus through this thing called hyphal anastomosis. And I'm not going to go into it, but essentially it's when all of the tubes that make up the fungus are able to fuse and they can share their cellular contents, including this virus. But that is hindered by the fact that there are 64 different strains of Cryphonectria, and only individuals of the same strain can spread virus to one another. So type 1 and type 1 can give each other virus. But if you have a Cryphonectria isolate that's type 1 and one that's type 22, they couldn't spread the virus to one another. We are working on that at West Virginia University. Dr. Nuss at the University of Maryland created an individual with no strain type, so it can give this to it. This chestnut blight individual with no strain can give the hypovirus away pretty freely to other Cryphonectria individuals, but again, I'm not going to spend a lot of time on that because it can get kind of funky and confusing. So what I need you to take away is that the chestnut blight fungus is very debilitated by these hypoviruses, but we're working on ways to better spread these hypoviruses in the forest. So here at WVU, we do this a lot. We use hypovirus to preserve the American chestnut as well as we can. And these are all cankers that have been treated with hypovirus. They look pretty nice. Here on the left, this guy is creating some callus tissue, and it is kind of stopping this fungus from growing. It's growing so slowly that the tree is able to create this callus and kind of wall off the infection. Here in the middle and on the right, these are both cankers that have been healing for a while. And we can see this is the outside of the canker out here. And this is the outside of the original canker out here. But we see this plant material, this tree, is healing over the edge. And eventually these two sides are going to meet and the tree is going to cover up that hole where the canker was. And you'll never be able to tell it ever happened. If you cut the tree, you'll be able to see a dead area on the inside, but you won't be able to see from the bark. It's just like a lawnmower cut or something like that. And the way we do this is we treat the cankers using a very, very, very scientific method. Are you ready? We're going to take a bunch of fungus that is infected with the hypovirus, of the chestnut blight fungus infected with hypovirus. And we're going to throw it in a blender with a bunch of water, blend it up, and put it in a squirt bottle. Then we're going to go out in the woods. We use a leather punch and a hammer to create wounds around the margin of the canker, which you can see here. And it happens just like this. Tink, 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 tink. And then we have a bunch of holes in our canker. Then we use a squirt bottle to put our slurry, which is what we call our blended up fungus, into all of these holes. And then we spray slurry all over the top of the canker and use just a paintbrush from Lowe's to paint all of that slurry over the top. Then we cover it with a laboratory pad and tape it down with masking tape. So even though there's a lot of complicated research that goes into getting to this point, we make a lot of effort to make this something that you don't need a lot of specialized laboratory equipment or specialized training to actually use. We try to make it as accessible as possible to general landowners who may want to use this. So in conclusion, here's what we know. The American chestnut, or Castanea dentata, once dominated Eastern American forests until the chestnut blight fungus, or Cryphonectria parasitica, was discovered in the early 1900s. And something that I actually forgot to say is that the chestnut blight fungus eats chestnut trees by breaking them down with acid. So all of these points where we were talking about, with this picture where we were talking about how the tree is breaking it down, or how the fungus is breaking down the tree, excuse me. The fungus is creating this acid known as oxalic acid, and it's using that to kill the tree's tissues. And the reason that it affects American chestnut and not Chinese chestnut is that Chinese chestnut lived with this fungus for so long that it was able to create a mechanism to break down that acid that's killing it. But the American chestnut never experienced anything that was producing oxalic acid to try to kill it, so it never had to create this toolkit to survive. So when chestnut blight was introduced to the US and it's using oxalic acid to break down the tree, the American chestnut just didn't have a way to combat it whatsoever. And that's what our lovely colleagues at SUNY ESF are working on with the transgenic trees. They're using an enzyme which breaks down oxalic acid, putting it into the tree, and that allows the tree to survive infection. Okay, so the chestnut blight fungus eats chestnut trees by breaking them down with an acid. Hypovirulence, which is caused by the fungus becoming infected with the virus, can allow a lot of trees to survive chestnut blight. And that's because the virulence of the fungus is reduced. It's white, so it's easy to tell, luckily, that it is a hypovirulent isolate because pigment production is reduced. Growth is reduced and sporulation is reduced when infected with the hypovirus. We also have barriers to using hypovirulence, but researchers across the Eastern US are working to resolve these issues and hopefully allow this to become a more widespread use for landowners with important chestnut trees. So with that, I will take any questions you have.
Thanks so much, Amy. And we have a lot of questions.
Ooh.
Yeah. And I want to, and I'll also, your predecessor, Mark Double, is here. Hi, Mark.
Hi, Mark.
And he said chestnut and oaks are both in the Fagaceae family. The chestnut blight fungus attacks readily American chestnut, as shown by the cankers on Amy's slides. Oaks, mostly post oak, can become infected by the chestnut blight fungus, and it presents itself as butt swell rather than cankers. While the fungus can sporulate on oaks, it does so significantly less than on chestnut. And we had a couple, the reason he wrote that is because we had a couple of questions about why it affects chestnuts and not oak. I don't know if you can elaborate a little bit on Mark's answer. Do you have anything else to add to that?
I think that's perfect.
Okay. I don't know.
Thanks, Mark.
I know. I don't know. Maybe you can't answer this, and my knowledge of plant pathology is pretty remedial. But what about host specificity and how a host recognizes what a pathogen is versus a pathogen recognizing, hey, I can eat you? Is there any sort of that thing going on between oaks and Cryphonectria and chestnuts? What about that recognition system between a host and a pathogen?
I believe so. I can't tell you specifically what the oaks are doing because I actually haven't looked into that. But yeah, there is a lot of host specificity there, and it's not a coincidence that chestnuts can bother most things in Fagaceae.
Yeah, and so Frank asked something similar. You know, oaks and chestnuts are very closely related. So what is it about chestnuts that makes Cryphonectria want to eat them and not oaks? And I think, you know, we don't really know the underlying mechanism other than the chestnut doesn't know how to how to fend it off, or the pathogen recognizes the chestnut and not the oak as something that it can't eat.
Mhm.
Let's see, Mike Alcott says, do we have any idea of the relative numbers of blight spores present in the woods where there are scarlet oaks or other host species, but few if any chestnuts? In other words, does the fungus actively sporulate and disperse well in the absence of chestnut?
Not well, so I can't tell you exact numbers. I actually think Sandy might know that because I mean, every time I have a question, she ends up having a paper on it, which is incredible. But yes, so the chestnut blight fungus doesn't do as well on oaks. It's not going to produce a ton of spores, but it is producing enough to still pose a significant threat to any chestnut region that would be in the area.
And so I'm glad you mentioned Sandy Adignostaki. So Dr. Sandy, for simplicity, will be joining us in February, so February 19th. Sandy and Michael Nave are going to be talking to us about chestnut cultivars. If we had questions about hypovirulence, I'm sure she would answer them. And also to follow up too, I'll mention this again, but in April, we're going to be joined by Mark Double and Laurel Rogers. I see Laurel's on here too. Hi, Laurel. Elaborating on the great foundation you gave us, Amy. We're going to talk about cruddy bark and a little bit about what happens to cankers as they age. So there's way more to talk about this, and we're going to devote a couple more chats to it. Tyler Payne asks, have any European chestnuts or have European chestnuts been as devastated by the blight as American chestnuts?
No. So there are several reasons for that. One is that European chestnut is very marginally more resistant to chestnut blight than the American chestnut is. But number two is that hypovirulence works a lot better in Europe. And that's not something that I was able to spend a lot of time on, but remember I talked about how in order to spread the hypovirus, individuals of the chestnut blight fungus have to have the same strain type. And here in the US, we have all 64 strains, but in Europe, they don't have quite as much genetic variability. So they have fewer strains of the chestnut blight fungus. So it's easier to spread hypovirus in Europe than it is here in the US.
Thanks, Amy. And I see Ursula Heiniger is here. She and hi, I'm sorry to give all these shout outs, but Dr. Heiniger, I know, studied chestnut blight in Europe, and I think Switzerland primarily. So I don't know if she might want to be able to elaborate in some of the questions or chats on some of those questions about spread in Europe.
Yeah, that's awesome.
So let's see. We've got Anita Klein asks if I lost it. There's so many questions coming up, I keep losing them. Anita Klein,
no.
If a tree has been treated by the hypovirulent strain of chestnut blight, is it resistant to a subsequent infection on a different part of the trunk?
No. And that is something that we struggle with. And I'm going to tell on myself, our entire lab is really short. I think the tallest of us is maybe 5'8". So we do have a little bit of trouble when we're treating because we can't reach some cankers. So if a canker has been treated with hypovirulence, that canker is very likely going to be debilitated and the tree is going to survive that particular infection. But because the virus cannot live outside of the fungus, it's not something that's being transported around in the tree. So if you get a canker in the canopy and we've treated at 5 feet, the canopy canker is going to continue to kill the tree. We're actually about to initiate a study in our orchard where we're going to use orchard ladders to get up to these tall cankers and see how much effort and time that takes as opposed to the positives. But yes, in a forest setting, it's a little difficult to keep trees alive above where you can reach them.
Well, I think similarly, so Balfour Sartor and a couple of other people ask, what about it spreading to other trees? If it can't spread within a tree, or it's very difficult to get that to happen, what about it spreading outside of a treated tree?
So it can spread to a point, but it doesn't spread incredibly well. So trees that have chestnut blight that need to be treated with hypovirulence will need to be treated routinely. It's like a pesticide, or think of it like a pesticide treatment where you have to spray every year or 2 or 3 years, and trees that you don't specifically treat aren't going to be receiving that treatment.
Well, I think this goes along. I have several questions on here from folks who ask about it's sort of they're questioning about like an immune response. If you infect it with a hypovirus, does that then protect the tree in any way from subsequent infections?
No, it doesn't.
Yeah, unfortunate. I got a couple people ask about spreading the hypovirus. Can you use a drone or fog this somehow from the air and make that an easier way to spread things? Thought about that at all?
I wish. I have thought about it because I come from a forestry background where we do a lot of aerial sprays and backpack sprays and things like that. But no, because it has to live inside of the fungus, and because I didn't mention this, but we have done studies where we didn't wound the bark of the tree when we treated it, like I showed with the leather punches, where we just put our inoculum slurry over the canker, and that was not effective because we are hoping to be able to utilize that. We've also thought about coating like shotgun pellets, but we're afraid that they would get too hot. So, you know, there's a lot of crazy ideas we could, or not crazy, but fun ideas that we could look for, but unfortunately none of them have worked so far.
Lot to study.
Yeah.
So Craig asks, and an anonymous attendee also asks, can we get this stuff? How do we get hypovirulence to treat our trees? Is it going to be sold? How do you get access to this stuff?
So that's a tough question because we are currently, thanks to Don Nuss, and I really can't shout him out enough. We have this individual with the chestnut blight fungus that we call the super donor, which can donate hypovirus to any strain of the chestnut blight fungus. And we are working on deregulating that right now. We've run into some issues, so it's gone back and forth about whether or not we think it'll be deregulated, but we are working on it and hopeful to be able to give those to landowners. But as of right now, your best bet is probably to send me some bark plugs, and I can give hypovirus to your isolates and send them back to you, and you can use those. But without a lot of lab time and without using the super donor, it's difficult for people who aren't able to use this.
Am I correct in saying that in Europe it's sold? Is this like I've heard, I've never seen, but it's sold in toothpaste like tubes to orchardists that they can use to treat trees commercially? Can you substantiate that myth?
I have no idea. I've never heard of that.
Okay, I think
so. That's awesome.
But just because it's so much easier to spread in Europe, and I have a couple questions about that. I do have an answer here. It says to Dennis's question, it is correct that the hypovirulent fungus is at a disadvantage. Oh, this is a different question, but I'm going to answer it anyway. It is true that the hypovirulent fungus is at a disadvantage on American chestnut. The virus-containing strains do not spread well from tree to tree in the US, but that is not the case in Europe. or many areas of Europe, the slightly more resistant European chestnuts do have an advantage over American chestnut. And if you guys get a chance, go back into the TACF magazine, the Journal of the American Chestnut Foundation, the Chestnut magazine. There was a great paper that Mark Double wrote with Bill McDonald. Amy, were you on that paper where they talked about the marriage experiment there at WVU?
I wasn't on that one.
So that's a nice overview talking about how the trees that had more resistance did show greater reaction to the hypovirulence. So it looks like if you treat a susceptible tree, yes, it helps over and above if it was, not resistant at all and didn't have any sort of help. But the more resistance you have, the more efficient the hypovirus is at protecting that tree.
Yes. Yeah, so you could think of it as an infection on anything. If you're resistant to it, slowing down that growth even a little bit can help, especially if you're resistant. But if you're very susceptible, you need to slow down the growth a lot
more
to be able to survive.
Right. And Ursula, thank you. She says it was sold in France, but probably not anymore. In Switzerland, the hypervirulent strains were custom made by her former institute. So kind of like what you were suggesting, Amy, making customized strains to that cankers that you have.
I love the toothpaste idea.
Isn't that cool? I always have this vision of like chestnut growers going out and, squirting it on some cankers. So Mike Alcott asked a question that I have quite often. There are some relatively large chestnuts that appear blight-free in regions at the periphery of the former range. For example, the Allegheny National Forest, Maine, Vermont, Wisconsin. Is it likely that the blight itself is present in such areas at low levels? Why are they surviving? Is the blight there? Is it going to get there? Talk to us about spread.
So I think there's a lot of debate about that. In my opinion, I think that it's just that it's so far on the edge of the native range that there's such low levels of blight that they're escaping. Because I will say we did a lot of research in Wisconsin in a stand of chestnuts that had been planted by settlers that took chestnuts with them as they moved west. And these trees were very, very large and living until chestnut blight came into the area. And that's the picture I had earlier with those dead standing trees in the forest. Actually, if I can go back, this is in Wisconsin too. These are some of them that are still alive so far, or they were last time we were there. So it's my opinion that the ones on the edge of the range are escape trees. And it's unlikely that they're actually resistant, but that doesn't mean they're not still valuable. And that doesn't mean that we can't still evaluate more closely their resistance.
But is the blight there, like on the periphery? So I've always said, and correct me if I'm wrong, that especially these northern parts of the range, like the Allegheny National Forest in Northwest PA or Vermont or Maine, where you have big trees, 3-foot tall trees, yes, they've escaped, but it's, I've thrown out there that the blight doesn't like it in the north as much. It's not as warm. The blight, can't, it lives, but it's not as happy as in the warmer south. Is that true, or am I spreading misinformation?
I'm not sure that I've looked at that very much. I do know that, Savage River State Forest in Maryland isn't Wisconsin, but it is colder there than where a lot of our studies are. And our cankers grow okay during the winter, but not extremely quickly. So I could get on board with that theoretically, but I don't know the facts, is what I'm saying.
Same here. Someone should look at that. All right. So Linus Schmitz asks, do hypovirulent isolates produce less acid to break down the host tissue?
I don't think that that's why they grow more slowly. I think that they're just sick and they're inhibiting growth. Because even when I grow them on a plate, a nutrient auger plate that just has a bunch of yummy stuff that's ideal for fungi to eat, they still grow a lot more slowly. So I'm not sure that I know the exact levels of oxalic acid they're producing, but I do know that they always grow slowly. So I would not say that that's the reason.
Okay. We still have a ton of questions, Amy.
Yeah,
So
I love it.
yeah, it's awesome. You guys are amazing. Are hypovirulent cankers orange? Number one. Number two, how is cruddy bark different from hypovirulence, if at all? And so I'm going to repeat, in April, we're going to talk way more about cruddy bark. But I'm going to go ahead and ask you, are hypovirulent cankers orange? And what's the difference between cruddy bark and hypovirulence?
Man. Okay, so I don't know what to say about cruddy bark because I don't know a lot about it. But I will say, hold on. I'm trying to get you a picture so it makes more sense. So fungi grow radially. They are similar to a lot of plants where all of their growth is happening on the outside. Once they grow in the middle, that's just kind of set. So when you have a canker like this one on the left, see these orange dots? That's still fungal material, so it still looks orange. So when we put hypovirus into this canker, the resulting growth from then on out should be white, but all of the stroma that you're going to see popping out is still going to be orange. So a hypovirulent isolate, and you see this on a plate too. All of the growth after it's been infected will turn white, but the growth previously will say orange because it wasn't infected at the time. And it's not producing, it can't go back and take the pigment away, but it no longer produces pigment from that point on. Does that make sense? Okay, so it should still look orange on the tree, but you would have to sample it in order to see the change.
When you played it, it looks white. And I have a question from Hill Craddock. Hi, Hill.
Hi.
He asks, are you going to talk about the super donor? And the super donor, from what I recently learned, is brown. Is that correct? And it's kind of a neat thing.
Yeah, the super donor is brown. And that was strategic by Dawn. There are Dr. McDonald, who has recently retired. They discovered, him and Mark discovered some brown chestnut blight isolates a long time ago, and those were used to create the super donor. So it's easy to visually tell if it's a super donor because it's still brown rather than with an orange tint.
And then where are we with the super donor? I think you might have covered this, but I have a few questions about, that. I mean, is it still being researched? Is it going through deregulation? Anything you can talk about in those aspects?
We're still hoping to deregulate. It's still being researched. We're actually working on a permit with Andy Newhouse and some other people with TACF to do more research here at WVU using the super donor in an orchard setting, because it seems like that's the best place for it. But we don't have any information we can give you as far as like a time you might be able to expect it to be deregulated, or if that's going to happen. I can just tell you we're really working on it because we want to be able to get this into the hands of landowners.
Great. So Bruce Levine, hi Bruce. What
Hi.
can you tell us about the hypovirulent fungus you introduced to the forest with your inoculations? So what can you tell us about whether the hypovirulent fungus you introduced to the forest with your inoculation spreads in the forest?
Oh, it does not. So we have used super donor, which is a genetically modified one, and it was under permit. So all of my research is focused on the super donor. And we have sampled surrounding trees, and I've sampled hundreds of cankers that I put super donor into, and I have not recovered super donor except 2 to 3 times. So it doesn't even persist in the canker very well. It hangs out there long enough to give the hypovirus, and then it's kind of overwhelmed by other chestnut blight individuals in the canker. So from what I've seen, it does not spread very well. I believe from what we saw in Wisconsin with spread, hypovirus wasn't spreading very well. It does spread to a degree on its own, but it's not something that you can inoculate a stand of trees and then expect the rest of your woodlot to be protected.
Sorry, Amy, I muted you by
Sorry.
accident. I was trying to mute me and I muted you.
Done.
Yeah. So Roger Wilby and a couple other folks in the north say that blight grows well in Maine and has killed a lot of trees. And yes, the blight does still grow there. I don't want to deny that at all. Let's see. I had a couple people ask whether this is going to be available afterward. Yes, we're recording this. It's going to be up on our Chestnut Chat website. The latest chat in the chat is a link to the Chestnut Chat series. Jules who is on here. Thanks, Jules, she downloads, she reviews everything, and then puts it up into YouTube. So many thanks to her for getting all of these up online for you guys to watch after the fact. I think we have time for a couple more questions if you still have some time, Amy.
Oh, I'm down. Yeah.
Okay. Wilburn Price asks, would a limestone solution kill the acidity if it was sprayed onto a canker?
I have never heard of anyone trying that. I don't think so. I'm going to be honest.
I don't
Because the fungus would just continue producing. It might neutralize it for that day, but the fungus is going to keep producing acid.
Well, and that happens under the bark. I mean, before you can see it. So if you ever cut into a canker, and I recommend it, it's a good party trick, cut very lightly into a canker. And what you see on that canker is not the extent of the fungus growing. Do you have a good picture of that, Amy? Yeah. Perfect. Excellent. And so if you do this on a tree, you'll see that the mycelium is extended under the bark. And so if you sprayed anything onto the tree, the fungus is protected by the tree itself from anything that you would spray on it. Now, I know folks have tried spraying alcohol on cankers and have had limited success, but you have to keep spraying for this same reason, because the fungus is kind of hidden under there and protected while it's producing acid, killing the fungus, eating it, and then you see, the sunken appearance that you see, more centralized at the point of infection. All right.
That's a good point, Sarah.
But it is a fun party trick to kind of see how far it's actually going.
Mm-hmm.
Scoville says, has hypovirus been registered with the EPA as a microbial pesticide?
No. So the hypovirus itself is found wild in the US. I believe one of them's from Michigan, and then another one was found in Maryland. So they're naturally occurring, but the super donor, which is something different, that's a genetically modified fungus that is regulated, but it's not registered as a pesticide yet. We're working on it.
Dawn asks, is hypovirus invading a fungus comparable to a phage virus infecting a bacteria?
Honey, I'm sorry. I'm a forester. I do not know, and I wish I could tell you.
Maybe Mark or someone would
Yes.
Speak to that. Andy Newhouse he said at least some of the differences in hypovirulent strains are likely due to reduced oxalate production, and Sandy would know more. So maybe I'll save that question for February when Sandy's with us.
Sandy's always, if I don't know the answer to a question, I'm always just like, Sandy would know that.
Sandy knows. That's for sure. She's definitely the chestnut queen.
Mm-hmm.
Mark says the phage virus infecting a bacterium is similar. Thanks, Mark.
Thank you, Mark.
David asks, do viruses specific to each of the known blight subtypes exist? Is there a virus repository that researchers use?
Can you say that again?
So there are 64 strains, right, of the fungus. Are there viruses that are specific to the strains? And if so, is there a virus repository or a library where you can get viruses?
There are not. I have never seen anything saying that certain viruses work better on certain strains. There are a variety of viruses. There's actually 4 large group of viruses, and we typically use what's called CHV1, which is Cryphonectria hypovirus 1. And we do have a variety of viruses that we keep. We have a bit of a repository, but I'm not sure that there's anywhere that they're really available, because they can't live outside of the fungus. So anywhere that you have the virus stored that's living and is going to be able to be used is going to be stored inside of a chestnut blight fungus individual that has a strain to it.
I'm going to take 3 more questions. So I'm going to take another five minutes of your time, and the audience will bear with me. And we're not going to get to all the questions. So if we don't get to your question, we'll answer it via email. But there are a couple really good questions here.
Yes.
So have the hypoviruses been sequenced?
Yes. And Dawn and Brad Hillman would certainly know where you could find that. But I myself have extracted the virus and have bits of it sequenced and found them on NCBI.
I had someone ask this request that I have you answer this question. Dennis Ham asks, and I think this is a toughie, because of the direct treatment requirements and the fact that all 64 strains are here, what is the ultimate value of this technique, or is it just about understanding the limits and vulnerabilities of the fungus?
I think that the ultimate value, I think it's something similar to antibiotics or something like that. I think that we know that there are major limitations, but I also think that we know enough about it, but we don't know everything. I think that there's just a matter of time until we find a way to really be able to use it widely. So right now it's just about trying to keep orchard trees alive, or especially important trees alive, especially if you have maybe a large escape tree that's been infected, or a tree with very good genetics that you just need to hold on for another 20, 30 years, something like that. It is difficult, and it's something that's very time and labor intensive, but I think it's also a matter of time until we figure out how to make it more widely accessible and more easily applied.
And if I might add, I think there's 2 other things to add to that. One is the commercial chestnut industry. So we had a few months ago, we had, I can't remember her name. She joined us from Michigan State. There's a huge chestnut industry in Michigan and Iowa, burgeoning in Ohio, Pennsylvania, and a lot of people use Chinese chestnuts, but some are using European or Euro-Japanese hybrids. And especially those European hybrids, which are susceptible, they could really benefit from, and it is more labor intensive than on a landscape scale, of course, which it's not really effective yet. But this really going to help folks who are planting any sort of susceptible chestnuts for commercial production. I've heard, we're all dealing with COVID, of course, but whenever anybody talks about, whether masks work or vaccines or whatever, you can debate the efficacy of any single technique. But I think with chestnut blight, as with any sort of security measure, I love the analogy of Swiss cheese, the Swiss cheese model of security. And so like resistance, gene editing, biotechnology, hypovirus, like the more of these slices of Swiss cheese you line up, the fewer and fewer holes go all the way through. And so I think that hypovirulence is just another slice of Swiss cheese that you add as another layer of security to help protect these trees. And I think I agree with you too, Amy, that eventually we'll find a method that allows these to spread more readily. But I think in the meantime, it still has merit, even on the limited application, and maybe not on a landscape scale.
And I'll add, it's just like masks and social distancing and washing your hands. None of them work totally by themselves, but together you have a much better chance.
and they don't solve it entirely, you know, so, but each layer helps. Amy, thank you for your time. I'm going to cut it off there. We have a lot of other questions. So Amy, I'll probably get in touch with you. Kendra too, I want to give props to Kendra. She has been madly typing in question or answers to these questions that we haven't been able to get to. Thank you guys for your interest. Again, I'm going to repeat February 19th, we've got Dr. Anna Gnostakis and Michael Nave. They're going to talk to us about cultivars. And like I said, I'll probably pick Sandy's brain for some of these hypovirus questions. April, we've got Mark Double, who has been prominent answering some of these questions, and also Laurel Rogers. They're going to talk to us about cruddy bark and the complexity of cankers. They're really interesting what all happens in these things. So we definitely have way more to talk about blight. Obviously, with all these questions too, there's way more to investigate about blight than we were able to get to today. Thank you for joining us for our first edition of Chestnut Chat in 2021. Amy, thanks. I'm going to give it to Lisa, and then maybe she can see if you have any final words.
Well, you're getting a lot of props in the chat room, Amy. Great job. Thank you so much. We really appreciate this enthusiasm, and I'm telling you what, I think you have some job security with this project for a while. It's going to take some time, right? All of us. So I'll just make a quick goodbye to everybody. I just saw this at the office this week, that brand new chestnut coming out, so you should be getting that in the mail. Sarah did a really nice article on the Chestnut Chat, and shows what next ones are coming up. So we're really excited about that. There's a wonderful profile of Mike and Louise Alcott, our wonderful volunteer spotlights, and I think it's a great issue for Chestnut. Jules, thank you for getting these magazines out practically single-handedly, although we do have a lot of wonderful guest authors in here as well. So Happy New Year again. Everybody stay safe, and I hope we'll see each other sometime this year. So take care. Thanks, Sarah. Great facilitation as always. And Kendra, take care, everybody.